In this lesson, we're going to learn how to approach a planning question. A planning question will appear in your practical exam. So your practical exam usually would comprise of 2 to 3 experiments and 1 planning question. And the planning question requires you to design an experiment and to state the steps, the procedure required in order to achieve some objectives. For example, to find the amount of calcium carbonate present in seashells. Alright, so to approach a planning question, there are certain considerations that you must take. These are the 3 recommended considerations that you need not write in your answer, but it will be good for you to think through before you start writing your answer. Okay, and these 3 considerations are the aim, your approach, and then the chemicals and reagents. Right? Later, when we look at an example, it will become much clearer. The aim of the experiment is usually stated in the question. So the question tells you what you are trying to achieve. The approach would be what kind of experiments can you conduct in order to achieve the experimental aims. And for certain questions, it will require you to identify 3 variables. Okay, the 3 variables are independent variable, your dependent variable, and your control variables. Now, for those of you who cannot remember what these 3 variables are, later when we look at an example, I will elaborate the 3 variables again. And the next thing that you need to consider would be the chemicals and apparatus required. By now, you would have done a range of experiments, and it will require you to recall when you were doing that kind of experiment, what are the chemicals and apparatus that are required. For example, if you are doing a titration, you will definitely require an acid, an alkali, an indicator, deionized water, and then the apparatus that you require would be a burette, a pipette, a conical flask, and so on. Now, it will be good if you can be specific. So if you are using any solutions, to state the concentration, and if you are using any solids, to state the form. So for example, if you are looking at calcium carbonate, is it in powder form, is it in lumps, or is it in chips? Now that you have considered the aim, the approach, and then the apparatus and chemicals, let's look at how we can incorporate this into our answer. Alright, so from this point onwards, we are looking at the things that you should be writing in your answer. Right, the first thing that should appear in the answer for all planning questions would be your procedure. Okay, in your procedure, it is very important for you to clearly state the chemicals and the apparatus needed. Okay, so as mentioned in the earlier part, the pre-answer considerations, there's no need for you to write down in your answer. So in order for the markers to know what chemicals and apparatus you are using, it's very important for you to write them clearly in your procedure. Alright, and if an experiment involves the 3 variables, we should be able to infer very easily from a procedure what are the 3 variables. Again, when I'm going through an example later, I will emphasize on that. And for certain experiments, it is much easier if you draw a labeled diagram. Right, from the labeled diagram, you can show in just one diagram all the apparatus and chemicals that are required. So that saves you a lot of trouble in listing them out in your procedure. The next thing that needs to appear in your answers for most planning questions would be results and discussion. Okay, now that you have listed the procedure for your experimental approach, the next thing that you need to write down is what kind of data can you collect from the experiment, and more importantly, how can you make use of this data to achieve your experimental aims. Okay, so it can be in the form of a qualitative description, like how do you make use of your data to therefore arrive at your conclusion, or it can be a quantitative kind, so you show the steps involved for calculating the whatever that you need to calculate in the question. And lastly, the conclusion. So your conclusion must be very specific to the aim of the experiment, right? It must address the aim directly. Again, I will show you an example in a while. Now let's look at the 2018 planning question. In the 2018 planning question, we are investigating the amount of carbonate in shells. So the question states specifically that we are given 1 gram of cockle shells, and then we need to design an experiment to determine the percentage by mass of carbonate. So let's go through the 3 pre-planning considerations. Okay, firstly, the aim. What is the aim? The aim is to find out the percentage by mass of carbonate in 1 gram of cockle shells. That is the aim. Okay, what is the approach? How can we go about determining the amount of carbonate in seashells? Right, if you are stuck, a good starting question would be this: in our syllabus, what can carbonates react with? Okay, what can carbonates react with? And in the topic of acids and bases, we learned that carbonates can react with acids to produce carbon dioxide and water, and of course a salt. Alright, so based on the reaction, is there a way for us to find the amount of carbonate present in cockle shells? The answer is yes. Alright, so since in the question they didn't mention what carbonate it is, there's no way for you to write a chemical equation, so we can only write an ionic equation. So we know that carbonates can react with acids to form carbon dioxide gas and water. Alright, so in this case, since we are producing a gas, we can actually collect the gas, determine the volume of gas, volume of CO2, and which will allow us to find the number of moles of CO2. And from number of moles of CO2, by comparing mole ratio, we can find number of moles of carbonate. And from number of moles of carbonate, we can go on to find the mass of carbonate, which will allow us to find the percentage by mass of carbonate. So this is the approach to the experiment. Lastly, we need to consider the chemicals and the apparatus. So over here, we need to react carbonate, we need to react the seashell or the cockle shell with acid, and then we need to collect the gas, and we need to be able to measure the volume of gas collected. Alright, so in this case, in your syllabus, you shouldn't be unfamiliar to experimental setups like this. We need to collect a gas. Okay, so we need to have a conical flask connected to a delivery tube, and then connected to a gas syringe. Okay, so in your conical flask, we will need to add your acid, and then we need to add your cockle shells, and then we have a delivery tube connected to a gas syringe. Okay, so this is roughly the experimental setup and the things that are required, the apparatus that are required, conical flask. Okay, so a question that you need to think of would be this: what acid do you want to use? Can we use any acid? Now, whenever you need to react a solid with an acid, okay, I would strongly recommend that you go for nitric acid. Okay, this is not the only acid that's suitable, but this is the best acid for reasons that I'll explain later. Okay, so whenever you're reacting a solid with an acid, I would recommend that if there are no restrictions, to use nitric acid. Now that we have gone through the three considerations, let us look at how we can write the procedure for this experiment. So firstly, we need to state that we need to set up the experiment as shown in the diagram that we have drawn. Okay. Next, very important step for experiments involving large solids is that when you throw an entire shell into acid, the reaction may take a long time. Alright, reason being only the shell on the surface is able to react with the acid. So in order to make the reaction faster, we need to break the shell into smaller pieces, and to do that, we need to use something called a mortar and a pestle, okay, to break it into smaller pieces. Alright, and then we need to transfer it into a conical flask, and to the conical flask, to the solid in the conical flask, we need to add excess nitric acid. Alright, the numbers given over here are just rough values. Okay, if you need in a detailed planning question, by right, you are supposed to roughly calculate the amount, the number of moles of carbonate present, and therefore to then calculate the amount of nitric acid that's required to react all the carbonate completely. Alright, but for a planning question at O levels, I doubt that they require you to perform the calculation unless otherwise stated in the question. So the only thing that's important is that you need to indicate to the examiner that you know that you need an excess of acid. Right? Why do you need excess of an acid? Is to make sure that all the carbonate is reacted, so the volume of CO2 is indicative, is proportional to the amount of carbonate present. Okay, so once you have added the acid, you need to very quickly replace the stopper, and then finally measure the total volume of gas that's produced. So this is the experimental procedure. In the procedure, if you take a look carefully, the apparatus are mentioned very clearly. We need to use a mortar and a pestle. Right, we need to use a measuring cylinder to add the acid to the cocker shells. Alright, we need to use a conical flask to contain the shells, and then we need to in the diagram we can see that we need a delivery tube, and then we need a gas syringe as well. Now for an experiment like this, there's no need for you to consider the variables, alright, because we are only doing one experiment. Alright, variables are only important when we need to repeat the experiments many times. So we will be changing something, and then we'll be keeping some other variables constant. So the next part, we'll look at results and discussion. So from the procedure, what data can we obtain from the experiment? The data that we can obtain would be the volume of CO2 collected. Alright, and then from then on, how do we make use of the volume to determine the percentage by mass of carbonate? For this will require a bit of your chemical calculation knowledge. Okay, percentage by mass of carbonate is actually equals to mass of carbonate divided by mass of the whole thing. Alright, mass of part over mass of whole. So this is the mass of shell, which is essentially just 1 gram times 100%. Alright, so importantly, we need to find the mass of carbonate in 1 gram of the cocker shells. And how do we do that? First, we need to use the volume to find number of moles of CO2. And then we need to compare mole ratio. Okay, compare mole ratio in order to find number of moles of carbonate. And then after finding number of moles of carbonate, we can find the mass of carbonate by multiplying by molar mass. Okay, and once we find the mass of carbonate, we can make use of the formula for finding percentage by mass, and that will give us a fictional percentage of X%. Now in the question, if you read it carefully, they require you to state any assumptions that are made. So if you look at this set of calculations, the very first one that we were doing to find number of moles of CO2 is to divide by 24 dm cube. Right? Why 24 dm cube? Because we have learned that the molar volume of a gas is 24 dm cube for any gas, but that is only true at room temperature pressure. Alright, so we assume, alright, one assumption that we need to make is that the measurements or the experiment is conducted under room temperature and pressure. Now one last part would be to write the conclusion. In the conclusion, I mentioned earlier that you must address the aim directly. So what is the aim of this experiment? The aim is to determine the percentage by mass of carbonate in 1 gram of cockle shells. So a proper conclusion would look like this, that the percentage by mass of carbonate in 1 gram of cockle shells is X%. Alright, do you need to calculate? Answer is no need. You just need to state, you have already shown the calculations, and then we just leave the final answer as X, and then we say that the percentage by mass is X. Now let's look at another example. Okay, so in this example, the aim of the experiment is to determine the most and the least concentrated acids. Okay, so in the description, we are provided with 3 acids, 3 solutions of hydrochloric acids, A1, A2 and A3, and then we are expected to design an experiment in order for us to find out which one is the most concentrated and which one is the least concentrated. Okay, so to begin, let us go through the 3 pre-considerations once again. So the aim of the experiment was very clearly listed in the question, that of the 3 hydrochloric acid solutions, A1, A2 and A3, we are expected to find out which one is the most concentrated and which one is the least concentrated. So for the approach, what kind of experiments can we conduct? Again, if you are stuck, it will be good for you to ask yourself these questions. In our syllabus, what reactions have we learned that acids can undergo, right? Or what can acids react with? Right. Another question that you can ask yourself, since we are looking at concentration, right? In our syllabus, what can the concentration of reactants affect? Okay, so if you can think of the answers to these questions, it will help you to conjure the approaches. Again, there are many approaches to such a planning question. There is no one correct answer. So if your plan works soundly, then it is correct. Okay, so first question that was asking, what can acids react with? We have learned that acids can react with metals, acids can react with carbonates, metal carbonates, acids can react with alkalis. Alright, so can we make use of any of this reaction to find out how concentrated an acid is? The answer is yes. Alright, so for example, we can react an acid with an alkali. This is a neutralization reaction. Okay, so how do we know which one is the most concentrated? Alright, we can perform a titration. This is the most straightforward one. we perform a titration, and then we find the title volume or the title value, the amount of alkali that's required. Alright, so the most concentrated one, most concentrated acid will require the largest volume of alkali. So this is the first approach that you can take. Secondly, we also learned that acids can react with carbonates, and we saw earlier that when acids react with carbonates, it produces carbon dioxide gas. Alright, so we can actually make use of that to find the amount of acid present by reacting the acids with an excess of carbonate. Okay, so in this case, it will produce CO2, and the acid that produces the largest, the most concentrated acid will be the one that produces the largest volume of CO2. Right, so this is the second approach. The last approach, or the last approach that I will be going through, I think there are many other approaches, right? But the last approach that I'll go through is this. So the second question was, what can concentration affect? Or in our syllabus, we have learned that concentration can affect the speed of reaction. Alright. So in this case, again, we can react acid with carbonate. So instead of measuring the total volume, alright, of CO2 produced, the final volume, we can actually measure the volume of CO2 produced over a fixed time interval, for example, over 1 minute. Alright, so what are we measuring now? We are measuring the speed of reaction. Okay, so in this case, the most concentrated acid will produce the fastest reaction. And how would that manifest? We will get the largest volume of CO2 produced in 1 minute. So over here, we can see that we have come up with 3 approaches. Again, I mentioned there may be more. All right, one other that you can think of is maybe to use an indicator, because the indicator will show the pH. That one is not very good for a few reasons, because sometimes when you have a strong acid like hydrochloric acid, even though the concentrations are different, they may produce roughly more or less the same pH, right? So maybe it will be just 1.1, 1.2, 1.3. All right, so how are you sure that the small difference would represent is not an instrumental error, right? So using an indicator, using a pH meter, it's not exactly an experiment anyway, it's just a measurement. All right, so over here, it will be good for you to design an experiment to show the most and least concentrated acid. Okay, so of the 3 approaches that I've listed down, which approach do you want to choose? Or which approach is more correct? There's no one approach that's the most correct. So the recommendation is this: choose the approach where you are most familiar with the procedure. Okay, that means you have done the experiment so many times, or you have written the procedure so many times that you know the steps very, very well. Okay, personally, I think for most students, the titration one would be the most familiar, because we have done so many titrations, right? So let us focus on approach number 1, and then we carry on with the last pre-consideration. Now, in the last pre-consideration, we need to think of what are the variables in the experiment. So let us revise once again the 3 variables. Independent variable. Okay, independent variable is the variable that we change from one experiment to another. Okay, so in this case, we are comparing the 3 acids. All right, from one experiment to another, what we are changing would be the acids. So it's A1, A2, and A3. This is our independent variable. All right, or if you look at it from a more generic point, it will be that we are varying the concentration of acid. Okay. Next, the dependent variable is the variable that we measure in each experiment. So in the first experiment, we are using A1, next experiment A2, third experiment A3. So what are we measuring? We are actually measuring the volume of alkali, or let's say that we fix it to be NaOH, the volume of NaOH required for complete neutralization. Okay, so this is the dependent variable. Dependent variable is what we measure in each experiment. And then lastly, control variable. What is a control variable? A control variable is something, the variables that we need to keep constant for each experiment in order for the comparison to be valid. Okay, what do I mean by that? So an example would be this. Number one, the volume of acid used. In this case, if we are doing a titration, in each experiment we are pipetting 25 centimeter cube of A1, A2 and A3. Alright, so if the volume of acid used is kept constant, then the volume of alkali required would therefore tell us the concentration of the acid. Okay, if we don't keep the volume of acid constant, then it will be a problem, because the just from the volume of alkali required for complete neutralization, we cannot make a direct conclusion. Okay, the other thing that's important is this, that the concentration of your alkali or NaOH used must be kept constant in the 3 experiments as well. If we vary the concentration, obviously the titer value would change, and the volume, the titer volume is no longer indicative of the concentration of acids. Alright, so for us to use the volume of NaOH required for A1, A2 and A3 to arrive at the conclusion of the concentration directly, we must keep, number one, the volume of acid constant in all 3 cases, and the concentration of alkali used in all 3 cases. Okay, many of y'all or many students will go on to write a third control variable, which is temperature. Now let's think about it. Is temperature a control variable? Is it important? When we vary the temperature, will it affect the titer value? The answer is no. Okay, temperature is only an important control variable if the reaction is a speed of reaction experiment. Okay, so for example, in approach 3 we are looking at speed of reaction, so temperature is a control variable, because if the temperature is not kept constant, then the speed of reaction may change due to temperature. Alright, so at this point we have gone through the 3 pre-considerations. I know it's a bit long and tedious, but that's because we are only learning at this point. So when you approach a question after learning how to plan, all these pre-considerations would just take a short while, and there's no need for you to write out everything as well. Alright, now importantly, what I need you all to do now is to keep in mind the variables. Alright, and in next we are going to look at the apparatus and chemicals required, and then later we'll see how these variables and chemicals and apparatus are clearly stated in the procedure. Okay, so now what are the chemicals and apparatus required? Let's think about a titration. So once again, in a titration, we'll require an acid, which is A1, A2, A3. We'll require an alkali. In this case, the most common one would be sodium hydroxide. We'll require an indicator. So the most common indicator would be methyl orange. And then the common apparatus for titrations, your burette, pipette, conical flask, white towel, retort stand, and so on. Okay, and right. Now let's take a look at the procedure now. Right, so we have mentioned a whole lot of variables, chemicals, apparatus, but take a look at the procedure. There are only 4 short steps. Everything is everything that we have mentioned in these 4 steps. Let's take a look. All right. First step, we fill up the burette with NaOH. Okay, so we have already stated that we need a burette. Then pipette 25 centimeter cube of A1 into a conical flask. So over here we state that we need NaOH A1 and then your indicator, and then titrate against NaOH until the solution turns from red to orange. Now this is the color change expected for methyl orange. For those of you who cannot remember, for methyl orange, in acidic solution it is red, when it's neutral it is orange, and then when it's alkaline it is yellow. Alright, so since we have acid in the conical flask, the starting color will be red, and the drop that changes it to orange tells you that we have reached the end point. Okay, so then after that we need to repeat steps 1, 2, 3 with A2 and A3. Okay, so let's check if we can clearly infer the 3 variables from this short procedure. Okay, from the procedure, can we tell that the independent variable is A1, A2 and A3? Answer is yes. Alright, because we are repeating the experiment with A2 and A3. Alright, so very clearly the marker would know that you know that the independent variable is A1, A2 and A3. Okay, from the procedure, can we tell the dependent variable? What are we measuring each time? Alright, we are measuring the volume of NaOH. Alright, so in this case, maybe we can improve upon this procedure by including this, that we record the volume of NaOH needed. Okay, so in this case, it's very clear that the dependent variable is your volume of NaOH. And the control variables, is it very clear in the question? Alright, yes, because we are repeating steps 1, 2 and 3. Alright, since we are repeating, if you look at steps 1, 2 and 3, we are using a pipette to measure 25 centimeter cube of acid. Alright, so the volume of acid is kept constant. And if we repeat steps 1, 2 and 3, essentially we are using the same NaOH, so the concentration is also kept constant. Alright, so from our procedure is very clear what is the independent variable, what is the dependent variable, and what are the control variables. Okay, and then as highlighted, the chemicals and the apparatus required are also clearly stated. Now we look at the results and discussion part of the answer. So from the experiment, this is a titration experiment. So most students will ask, do I need to draw the titration table? The answer is no need. For such planning questions, we need to draw a table that has the independent variable in one column and the dependent variable in the other column. So for example, in this experiment, the independent variable is the acid, which is A1, A2 and A3. The dependent variable is the volume of NaOH required for neutralization. Okay, is there a need for you to fill up with values? Answer is no need, but if you want to fill up, you can as well with realistic values. Okay, so for example, A1 would be maybe 25, A2 would be 18, and A3 would be 20. Okay, but again, there's no need for you to fill up with values if you do not want to. Okay, so the more important part is how then do we make use of this volumes, these titer values, for determining which acid is the most concentrated. Now, we have already discussed in the approach that the acid that requires the greatest volume of alkali is the most concentrated. But why? So the discussion part, you must state very clearly why. Why is it that the most concentrated acid would require the largest volume of alkali for complete neutralization? And for that, it must be very detailed, meaning you need to mention a few things. Firstly, since the volume of acid used is constant, that means in each experiment, in each titration, the volume of HCl or A1, A2 and A3 are kept constant. So the acid that's the more concentrated one would contain a larger number of moles of HCl. That's the first point. Next, if the more concentrated acid contains a larger number of moles of HCl, you will therefore require a larger number of moles of NaOH for neutralization. That's the second point. And the third point is this. Since the concentration of NaOH is kept constant in all three titrations, the more concentrated acid, which will require a larger volume of NaOH for neutralization, would therefore translate into a larger volume of NaOH since the concentration is constant. Okay, so with all 3 points combined together, we can see that the more concentrated acid therefore will require a larger volume of NaOH to reach the end point. Now, another way to explain how we can use the volume of NaOH to determine the concentration of acid is by using equations. Now, this way is more elegant, but you need to be more careful when writing it. So when we are looking at the reaction between HCl and NaOH, the mole ratio is 1:1, right? So therefore we can conclude that the number of moles of HCl is equals to number of moles of NaOH. And we have learned in chemical calculations that for solutions, number of moles is concentration times volume. Okay, so number of moles of HCl is the concentration times volume of HCl, and the number of moles of NaOH is the concentration of NaOH times volume of NaOH. Okay. Now, in the experiment, we have very clearly stated that the volume of HCl is kept constant, and also that the concentration of NaOH is kept constant. So if we replace them with constants, let's say K, it becomes very clear now from the equation that the concentration of HCl is directly proportional to the volume of NaOH. Okay, so by just showcasing this equation, we can summarize the 3 lengthy points which was described in words earlier by just one equation, one very elegant equation, right? So if you can write this equation, it explains all the 3 points already. Lastly, the conclusion again must address the aim directly. So the aim was to determine the most and least concentrated acids. So for the conclusion to address the aim directly, we must say that the most concentrated acid is, right, we can leave it blank. There's no need for us to indicate which one, because it required the largest volume of NaOH for complete neutralization. Okay, and since the aim is to determine the least concentrated one as well, so it will be good for you to write this. The least concentrated acid is the one that has that required the smallest volume of NaOH for neutralization. Alright, So what we have seen here is how we can approach a planning question. Firstly, the three pre-considerations. It may not apply for every planning question, but it will be good for you all to run through the three planning considerations before attempting to answer. So in your answer, you need to have the procedure. This is required for all planning questions, the procedure. The results and discussion, this one is required for most planning questions as well. And lastly, the conclusion. The conclusion must address the aim stated in the question.